Conor, I want to start today's episode with a little anecdote and a little pop science from a mutual friend of ours.
Okay.
In fact, she's more than a friend. She is the executive producer of Discovery Matters.
Aha, Andrea, what's she been up to?
Well, the one and only Andrea Kilin, and she'll tell us herself.
So me and my partner went to Amsterdam for a weekend.
They went there to visit some friends and watch a concert. And when they got there, her friends started talking about cryotherapy.
So we quickly Googled it. We found that it showed some promising results for people with chronic migraines, which is what my partner is unfortunately suffering from for the past almost 2 years now. We said, yeah, we tried everything from MRIs to CT scans to acupuncture and all the shamanism that you can think of. So let's go to cryotherapy. So we went there expecting nothing. And you get there, you're told what to do. You're given very fluffy socks. You enter a cabin with maximum 4 people. You get earmuffs so you don't get frostbite on your ears. You get gloves, you get a mask so you can breathe because you're going to go in minus 110 Celsius, which is minus 166 Fahrenheit. So in the first chamber, it's minus 50 degrees.
So this is basically the opposite of a sauna. You walk into a room where it's just freezing.
Uh-huh.
Just trust the Nordics to invent this stuff.
That's right.
Is it not cold enough in the winter?
You wear nothing but your undergarments. You go into a room where it's minus 50 and you spend 20 seconds there. You start getting used to it, to the cold. And then somebody knocks on the door, they go... And then you know you have to open the refrigerator door, the other refrigerator door. So we walked in and Alex started shaking like crazy. She started shaking really, really bad. And she said, no, I can't do this. I'm leaving. So I grabbed her by her 2 hands and I said, andas in, andas ut. We must know that in Swedish, that means breathe in, breathe out. And since my partner is half Swedish, I thought that might get to her faster. And she managed to focus on her breathing. We stayed the whole 3 minutes inside. And we're still alive. We felt very, very relaxed after it. It's a sense of calm overwhelms you almost immediately. We felt like we had a new body.
She went from a very cold freezer to a very, 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 very cold freezer. And this apparently resulted in a feeling of relaxation and revitalization. And Andrea said that those who worked there were almost addicted to this therapy.
And so would Andrea do it again?
Oh, yes, absolutely. I loved it. Alex barely survived. I'm proud of her. But if it was closer, I would go twice a week for sure.
So after chatting to Andrea, I thought, well, let's find the real science in this. Because in Discovery Matters, we love the real science, not just the pop science. And you know what cryotherapy does with cells and organisms is pretty important. I mean, we freeze them to take care of them in a way.
So getting away from the pop science of cryotherapy and looking at the real science and application of cryonics. I guess that's what matters in today's episode.
That's right.
Sunshine on my shoulders makes me happy.
Now, the notion of cold is usually kind of a negative. You know, we go on vacation to warm beaches, we think of the sun and we smile and we think of John Denver. But for some people out there, it is the complete opposite.
I think it almost depends on what your background is, because one thing that I've been looking at, not as part of work, but just in general, is looking at the sort of creation myths of different peoples, and it depends what they really hate. So a lot of the Nordic ones, hell is a very cold, icy place, and that sort of agrees with what you expect from very cold countries. The cold's a problem. But if you look at the other religions that come out, say from like the Middle East, like Christianity, hell is traditionally done as like a big fire, a very hot place. So I think it depends on really just what your background is. If you come from a very hot place, maybe you want somewhere colder because the heat's the problem. But for us in Northern Europe, often it's too cold, so we want somewhere warm. Not so much because it's warm, just because it's different and it's not what we have day to day.
And who is this?
So this is Peter.
Peter Kilbride.
And along with his colleague Julie
Meneghel,
we chatted about all things cryo.
The first thing that we have to remember is that human cells do not like the cold. Human cells are used to being in a very controlled 37 degrees inside their bodies for their whole life. And lowering your temperature, even by a little bit, your core temperature, can be extremely dangerous and just very uncomfortable to happen. It's what we see when we're shivering or go outside. In extreme cases, such as with polar explorers or mountaineers, you can see if their extremities get very cold, they can end up having a condition called frostbite, which is essentially the tissue freezing and then being permanently damaged. It never recovers even after rewarming. So I think our relationship with cold and what cryopreservation is, it's really the science of tricking cells into not realizing that they don't like low temperatures and then trying to use even some of the things that normally are bad for cells at low temperatures somehow to our benefit and to the benefit of the cells.
So what exactly is cryogenics doing to cells, and why is this cold good?
Temperature is going to impact on the rate of every reaction taking place in a cell. So the colder it gets, the slower all these reactions and diffusion rates will become, until a point where they stop completely. They will get to a halt. And this is where organisms will be in a suspended animation. At that point, they can be stored theoretically forever until temperature is warmed up again and biological reactions resume.
What is really common to all these organisms is the physical events that take place.
Well, hold on. Who have we got here?
That is Fernanda Fonseca, Research Director of the French National Research Institute for Agriculture, Food and Environment. And I chatted to her to get another side of the cryo story.
Our utmost ambition is to find general principles for rationalizing the cryopreservation of biomaterials, of cells, of organisms. So when you are going to cryopreserve all these organisms, you necessarily go through cooling, forming ice, and according to the cooling rate, you will have vitrification, and again back to higher temperatures, the reverse reactions, so the vitrification and going to a viscous state and melting, and you have your stuff again at ambient temperature. We have found out that there are still things to understand how it works inside the organisms, and that's our work today within cryopreservation topics.
Fernanda also told me about how she lives the science every day by reminding her kids that this transition of going from one state to another happens even when they warm things up in the microwave oven and when you have your breakfast. Talk about having a scientist as a mother.
I talk to my children on gas transition and vitrification, and they know that when they are going to the microwave with their bread, they are making gas transition. That's to give you an idea at which point that is a subject to vitrification that amazes me. And when you have your cornflakes in your box and it's still open for some days and it's not very crusty, you also had a glass transition that happened. When you have your candles in your mouth, you have glass transition and so on. So that is something that in cryopreservation, you work all the time with vitrification, the vitrification, going from a glassy state to a viscous state and the reverse reaction. And that is our life.
Okay, hang on a second. I might have to get me woolly jumper on or turn the heating up here because I'm starting to get a little bit cold. So hang on a second.
Just the thought of cold makes you cold.
There's just too much cold. I mean, okay. Right, that's better. So how do you make sure it's cold enough but not too cold and it doesn't destroy everything? Because, like I put sloes in the freezer to break their skins when I make sloe gin. And so why aren't we breaking everything when we freeze it so hard?
Well, I'd love to talk more about that sloe gin, but let's ask Julie about what you're talking about.
The problem with frostbites in human bodies, for example, this is related to the fact that here we are talking about tissues. So it's a 3D structure of many different cell types that are all functioning together. As this structure freezes, there will be ice crystals forming that may disrupt all this structure, even break cells. So this is something that we are trying to avoid when cryopreserving biological samples. So it is something that we can do, even if it requires work and optimization. We can manage to do this for cell suspensions, for a few specific types of tissues, but for larger organs or even whole organisms, this is not something that we can successfully manage to do today.
Okay, but the reality is, right now, it only happens on long space flights or in weird movies in Hollywood, correct?
That's right. That's right.
Okay, like Alien. That was a good one. They send the whole crew into space, they get cryopreserved.
All right, sweethearts, what are you waiting for? Breakfast in bed?
And it didn't turn out so well, did it?
So, yeah, the best thing about the movie's really good, right? It's the movie where nobody listens to the smart lady and they all die.
That's right. Oh,
It's a
listen
very
to the
simple
smart
film.
ladies. That's what we're coming
Yeah,
around
listen to the
to in this
smart
entire
lady,
episode.
or you will die. Yeah.
That's right.
So what do Peter and Julie think when they see scenes like that? I mean, so they're real cryobiologists. Do they just look at that and shake their heads and sit and think, well, what are we putting into the popular imagination here? Or, might it happen?
I think you have to follow on with what Julie was saying. In some respects, we can do organs to a limited degree. So if, say, you have a deceased donor, the organ can't survive in them for very long. However, if you remove that organ, whether it be like a heart or liver or kidney, and then transport it just above its freezing point, you get many more hours where it can be used for transplant. Still, it's not very long, and actually there's a huge number of organs that don't get transplanted just because physically, in the length of time it takes the, say, heart to get from the donor to the recipient, gets too far damaged. And I think that really one of the key aims in cryopreservation in general is there a way we can preserve whole organs for maybe even just a few days or a few weeks, and that way you could really properly match organs between different donors and recipients and have almost no waste in that process. I think when it comes to doing anything more than a whole organ, I think that's science fiction. It might be for a while. Each organ type and cell type has very different cold. You have to treat them in very different ways. It's science fiction for now, and I think it will be for a long time left. I think perhaps by the time you can freeze a whole organism, there will be so much more advanced science and biology, people won't really be dying in the first place. Perhaps it's that far away. So I think day to day, it's not something we'd work on.
Okay, since Peter mentioned humanity and preserving humanity, what does it look like for artificial insemination and preserving populations?
Well, in fact, that's actually where one of the first parts of cryobiology existed.
Cryobiology, its sort of biggest successes in reproductive medicine. So people say having IVF, they'll be able to freeze sperm, they'll be able to freeze eggs, they can store embryos. And one of the great successes of the past few years is people who are undergoing cancer treatment, which may then render you infertile. They can then take part of the ovary before treatment starts or samples before that, freeze it. So then people who historically would not have been fertile can then have children. And that's one of the big successes. It's also been moved a lot more when it comes to endangered species. So quite a lot of zoos around the world now, they have things called frozen zoos. And if they have an endangered species and say it dies, they'll take some tissue samples from that and cryopreserve it. And even if the species goes extinct, you've still got a small amount of biological material from them. And hopefully one day, perhaps be used to then bring back a species that's gone extinct in the period of time when many species are going extinct.
Okay, so what are some examples of this working or potentially working in the future?
Well, let's think about something ancient and massive.
Something ancient and massive. A T-Rex, Jurassic Park, yay!
Almost. There's a legend
No.
about bringing this animal back to life.
Are we talking about the woolly mammoth?
We are.
So the woolly mammoths is really quite a good example of how cryopreservation can work. So woolly mammoths existed in northern latitudes thousands of years ago before they were killed off by early human hunters. But luckily, because they lived in such far north latitudes, some of them were frozen just because it was the ice age, it was very cold. And nowadays, you can actually sometimes find woolly mammoths that still have intact soft tissue. Of course, the cells in that system are not alive. They're not something we'd freeze in the lab ourselves, but they do still have some DNA. So there has been some thought that we could extract that DNA and then use it to recreate a woolly mammoth. For example, perhaps by getting some DNA and then inserting into the egg and sperm of an elephant, which is one of the closest species currently alive that are like a woolly mammoth. I think that is something that I wouldn't say is impossible. It's certainly incredibly challenging because with species nowadays, you get sperm, you get eggs, you get the reproductive tissues that you need. When it comes to woolly mammoths, the DNA is normally very badly damaged. And the closest species to woolly mammoth is an elephant, which is actually quite different. And you need an elephant, you need a living uterus to have these animals reproduce. So I think that certainly it will be possible, and I expect that sometime in the future it'll be done. I think before that stage, we'll probably be looking at bringing back species that humanity has caused to go extinct just because we have much more material from them and because they were frozen using modern science opposed to just fortunately dying in cold climates where some of the soft tissue was preserved.
And you know, when I was chatting with Fernanda, she brought up the often forgotten side of cryobiology, and that is the environmental impact of cryobiology.
Until now, we were aware about quality of the products and the cost of our productions. And now we have to think multi-criteria approaches and consider the impact of the whole chain, the whole cycle of our product is having. And for that, we need, that is really amazing, is that we can not longer make research and work on research without many multidisciplinary approaches. So you need to have around you in this cryopreservation teams, biologists, physicists, biochemists, and mathematicians, and people modeling and people concerned by the environment, helping to assess the best strategy.
So if we bring it back to right now, what's going on today and patients, where exactly is this finding the most use? Where is it helping?
Let's go back to Julie and find out.
The area that has attracted much attention in the recent years is cell therapies, and especially CAR T cell therapies, chimeric antigen receptor. It is a cell therapy that has been developed for patients with some specific types of blood cancer. Their own T lymphocytes are being harvested from their bloodstream, genetically modify them and make them more stronger to attack cancer cells within their own body. They're then amplified to create billions and billions of them, and then reinfused back to the patient to help him or her fight the cancer. Cryopreservation is a critical step during this process because cryopreservation allows for more flexibility in terms of patient treatment.
And I think another area as well that's quite common and many listeners might have heard of will be cord blood cryopreservation. That's something Julie and I have been working on quite a lot recently as well. So when babies are born, you have the umbilical cord, and the blood within that contains a lot of early stage cells that can be very good for treating people with specific diseases later on, either the same baby who from the umbilical cord comes, or from other babies around. So one area which is a lot of science has gone into and it's still being improved is if you extract some of that early blood from the umbilical cord of newborns and then you can freeze it, you can then have very large biobanks with huge numbers of different blood from different babies, which you can then match to people who may need some sort of cell therapy in future. And I think that's quite a good example of where cryopreservation is really essential, because often in those cases, you'll need to store the blood for months, years, often decades before it becomes useful. And of course, you can never go back to a baby and get umbilical cord blood twice. When the baby's born, you have one shot and that's it. So that's one area where cryopreservation is really quite common now. It's something still an area of research where we try and improve the cellular outcome even further after decades of being stored.
It's just extraordinary, isn't it? That being able to reduce the temperatures to really, really cold is so fundamental to understanding things. Remember, we talked in our episode on quantum biology about needing very, very cold temperatures to see quantum effects, and that meaning that you wouldn't be able to see it at warm temperatures, and people thinking that didn't exist. So, like, being able to get things really cold is really important for science.
But I was also thinking about the episode we did about sense about science and what is real and what is pop science and what is beneficial for human health and advancing therapeutics. So I think this has been a really interesting journey from talking about Andrea and her experience walking into a super cold room and how that made her feel relaxed and rejuvenated to when we actually take cells and freeze them in order to rejuvenate them later and make them useful in therapeutics.
It's the kind of interplay between sort of what we read about science and what we want science to drive us towards and how we feel about innovation in areas like consumer health, and then people's imaginations. I mean, driving science forward. I mean, people who take science fiction ideas like the communicator in Star Trek and it becomes the mobile phone. And there's this really interesting interplay between pop science, science fiction, and real science and where the world can go. And it feels like we're in an age now where if you can imagine it, there's probably someone somewhere working to make it real. And that might be a good thing. And it could also be a pretty difficult thing.
So I think in the last about 20 minutes, we hope that this episode has answered some questions about cryogenics, but also raised some questions, because that's important that science is constantly raising the right questions. Am I right, Conor?
Yeah, you're absolutely right. Look, every day is a school day, and the best thing about science is being proved wrong.
Every day is a school day. We got there. Thank you for listening.
Thank you for listening.